Good afternoon for most folks. I'm Mark Schumacher. I'm the director of PARC. And for those that are joining us for the first time, we're offering a monthly academic seminar series. But first, just to make sure you understand that PARC is really our Pain and Addiction Research Center. It's a transdisciplinary center that spans both clinical and research communities, preclinical and clinical, both for pain and addiction science. There's a QR code that'll bring us to our website if you have an interest in finding out more or potentially, hopefully, joining us. Before we get to our speaker today, I just want to remind you that next month, Khaled Masawi, one of our own, is going to be speaking on neuromodulation for substance use disorder. He's an associate professor here in the Department of Neurology in our School of Medicine. And then finally, it's my real pleasure to introduce Dr. John Streicher. He'll be talking today about the orphan receptors, GPR63 and 153, enhance antinociception and pathological pain by suppressing microglial activation in the murine spinal cord. John is a professor in the Department of Pharmacology in the College of Medicine at the University of Arizona. His research program focuses on the signal transduction cascade of G protein coupled receptors, such as opioid and cannabinoid and adenosine receptors, in the context of chronic pain. This research focuses helping him design and test improved therapeutics for chronic pain without the side effects of current analgesic drugs like opioids. He earned his master's in neuroendocrinology from Oregon Health and Science University and his PhD in signaling cascades in heart failure at UCLA. John completed his postdoctoral training with Dr. Laura Bond at the Scripps Research Institute and then served as a faculty member at the University of New England until 2015. He's now then currently been at a position, as I mentioned, at the University of Arizona. Again, it's our great pleasure to welcome John, and I'll now switch and stop sharing and let him go from there.
Thank you so much, Mark, for the invitation. It's greatly appreciated. It's a great honor to be speaking with you all today. Thank you also to Anna for your help in kind of organizing this thing and getting it set up. None of us could do this without you. And so I want to talk today about some of our work. So, Mark just mentioned in my bio, opioid, cannabinoid, adenosine. We all know those names. But more recently, in the last few years in my lab, we've been pursuing a couple of orphan receptors and none of those familiar names and numbers that you know about. So I want to take some time to talk about that and talk about our work on these orphan receptors, brand new orphan receptors, and how we think they're regulating pathological pain. And so, there we go. And so to this audience, I'm not going to give the usual, pain is bad, opioids are bad, overdoses are up. We really should make alternatives to opioid therapies. I don't need to tell you guys that. You know it probably better than I do. But what I do just want to highlight briefly have been, and this is not exhaustive, of course, but some of the recent novel approaches to manage pain, both novel in terms of new regulators, new systems that regulate pain, both pronociceptive and antinociceptive, as well as new potential therapeutic strategies, right? So this is where I fit my orphan receptors into this sort of context. And so in the middle, we have our, sort of standard ascending to descending pain pathways system, which again, you probably all know better than I do. And just as a brief sort of a selection, a little bit of a smorgasbord or plate, entree plate, or sorry, appetizer plate of different approaches. We have things like, Ted Price's and others work to interfere with the translational reprogramming that underlies much of chronic pain. Same idea, except on the transcriptional side, using HDAC inhibitors and others to interfere with transcriptional programs that, again, underlie reprogramming in chronic pain. I'm going to be a little bit indulgent and include one of my other projects on this on this smorgasbord. I hope you'll forgive me for that. We've been looking at some novel compounds called terpenes and how they interact with the adenosine A2A receptors in the spinal cord, how that could be a sort of novel therapy, novel approach to regulate pain. New regulatory circuits like endocannabinoid and other ways to activate or bring those circuits to bear on reducing chronic pain. Of course, for this group, no surprise or to hear about neuroimmune modulation, the role of the microglia, astrocytes, and inflammatory cascades, neurogenic inflammation, and so forth, underlying much of the pathology of chronic pain and how we might be able to modulate that to improve outcomes. Selective sodium channels involved in transmission of pain, NAV 1.8. I've even seen a little bit on 1.7, 1.9. We have this recently approved drug, I practice this to see if I make sure I get it right. Suzetrigine, I think. Suzetrigine? I don't know. Maybe somebody knows. Newly approved NAV inhibitor for the treatment of pain. And so again, this is not exhaustive. And if one of you are working on a novel approach to pain, I do not mean to slight you by not mentioning your work in this slide, but just to sort of say that we're all working on these different ways and different approaches to investigate new regulators in pain, new therapeutic approaches, getting away from sort of the standard model that we know, as well as some of the standard therapeutics that we have on hand, which have their limitations, which you all know. And so in that context, I'd like to tell you then about our efforts to target orphan receptors. And so for a little bit of more information on this, so this is a pie chart, if you will, of the non-olfactory GPCRome. And most of them we know something about, right? So these are everything. These are your adenosine receptors, your serotonin receptors, your dopamine receptors, the metabotropic glutamate receptors. All those names that you know sort of fit into this chunk of the pie chart. But there is a sort of unexplored country, a great unknown, these orphan receptors. And so this number changes all the time. It goes down all the time, which is a good thing as we learn more. But last I checked, there are a little fewer than about 100 of what could still be called orphan receptors, where we don't know anything, or maybe we know very little about their physiological role and or endogenous ligands, endogenous roles in physiology. And so that's a lot, right? So this is a pretty big unexplored country, there be dragons kind of a location for us to plumb and to find new regulators, new GPCRs that could be regulating pain or anything else, right? So this is not only true for pain, this is true for heart failure and for kidney function, anything else you might think of. There's going to probably be these orphan receptors involved in there. And so we're going to go on. So what I wanted to do is go on this great exploration into this undiscovered country, find some new GPRs that regulate pain. And by saying it in this way, I don't want to come across like I'm trying to say that we're the first or the only ones to do this. Of course, we're not. Mark, you're one of them. And there are many others that have been also looking at GPCRs in pain and addiction and other areas. GPR3 and Alzheimer's, GPR6 and Parkinson's. We kind of go along here. 55, the new endocannabinoid GPR. We keep going along. Autism, and then we start to hit pain now. GPR171, GPR. Was it 16? I always get the numbers mixed up. It was either 160 or 162. This is one of the bad things when you have an entire series of proteins that are just a number. So it's hard for me sometimes to remember exactly which number's which. But those Daniele Salvemini, they had that. I think it was either 160 or 162 that they found in dorsal root ganglion. Anyway, I don't want to get too distracted. And so, others involved in neuropathic pain, different kinds of pain, inflammation, and so on. And so we are not the first. This has been done, and these new receptors are continually being discovered. New endogenous ligands for these receptors are sort of being discovered in an ongoing way, such as CART peptide, that was Salvemini's group. PEN, which was done by Lakshmi Devi's group, and so on. Right, so we're not the first, but we wanted to nonetheless tackle a new set of receptors and find something new out of this series. And so we started plumbing through and looking through the orphan receptors that were available. And unlike a lot of the time when you hit the literature and you want a new target to look at, we were looking for proteins that we didn't know what they did. So we didn't, if we looked up a GPR and it's got 30 papers on all these different physiology that it's involved in with the known ligands or whatever, that's not what we want. Maybe foolishly, but that's not what we wanted. We wanted something that was really new. And so by going through this literature, we're prioritizing GPRs with no established physiological function, no established ligands, either exogenous or endogenous, However, like we don't want to try to screen for GPRs that are, say only expressed in skin cells or something like that. Going to be a hard lift for something in pain. So we also wanted to make sure they were expressed in relevant CNS regions that could be potentially, at least to be feasible to be involved in pain regulation. And then you might say, well, wait a minute, if nobody knows anything about these ligands, or sorry, about these receptors, how do you know that they're in the relevant CNS regions? Well, again, I think you all probably know better than I do, but in case you don't, there are many RNA-seq and proteomic databases publicly available that you can look. And even if no one has ever studied your protein of interest, chances are it's been quantified in one of these databases. You can take a look at that. And so using these criteria, we settled on four GPR family members to look at: 63, 141, 150, and 153. And again, at the time, at least, essentially nothing known about any of these receptors and no ligands for them. This is the GTEx data set here, looking, at some relevant CNS regions, brain cortex, spinal cord, et cetera. And the levels detected are comparable, or sometimes even a little bit more so than, say, the mu-opioid receptor, which we use as a comparison to say that this is a relevant receptor. And so the expression levels are pretty good. And so thus established, we now want to screen these targets and evaluate if they could be involved in pathological pain. So how did we do that? How did we run that screen? So to do that, we relied on our CRISPR-based in vivo knockdown approach in wild type animals. So this is not like a knockout line, a germ line with CRISPR. This is not that sense. We take adult wild type animals that, we just get regular CD1 mice, and we're able to use these DNA constructs, complex not as viruses even, but with in vivo transfection reagent, inject them either intrathecally into the spinal cord or ICV into the brain, and get widespread tissue knockdown across these tissues. It's a really flexible and kind of easy to use approach. And it relies on a few things. Again, if all of you know CRISPR very well, I apologize for the redundancy here, but you have a few key elements. One is going to be this universal U6 promoter driving the guide RNA targeted to your gene of interest. Well, that'll become more relevant later. And then as well, a more standard protein open reading frame for the Cas9 that's being driven by, in this case, cytomegalovirus, which again is a universal high-level promoter in numerous tissues. As I'll get to later, we can change this promoter and change how the CRISPR knockdown construct works. And so we've done this before. We've published it a few. Here's an example of some data with a cell-selective approach versus just a universal knockdown. In green is AMP kinase, in red is CGRP. You see you have some of these yellow cells here where they're co-expressed. And then when we do our cell-selective knockout, we've lost specifically those yellow cells here in the CGRP zone, but not some of the others in the non-CGRP cells. So this is a really useful tool that we've used now for a number of projects and a number of papers. And then what that looks like then experimentally in terms of a timeline is you have your animals, run your baseline measurements, and then in this case, we injected all of the CRISPR DNA intrathecally, IT. So we started with the spinal cord as sort of a recent area of interest for us that's kind of easy for us to investigate. So we do these IT injections. You do them 3 days in a row consecutively, so multiple injections, and then you let these guys sit until day 10, right? So the 10 total days for the CRISPR-mediated knockdown to occur. And then interwoven in that 10-day, we put our pain model. And so starting on day 3, we do another baseline measurements, and then we start injecting paclitaxel. So this is a chemotherapy-induced CIPN peripheral neuropathy model. So type of neuropathic pain, again, you're probably all very familiar with it. Don't want to get too remedial here. And so that timeline is woven into the CRISPR timeline so that on day 10 of CRISPR coincides with day 8 of the CIPN, which in our model is sort of our standard test day. Then on that day, we again do our baseline measurements, inject in this analgesic, in this case morphine, and then we run that time course and see what that looks like. So that's sort of the overall model. Oh, by the way, I forgot to mention too. So if anyone has any sort of questions or anything's not clear, I'm a very informal guy, so please put the hand up virtually or ask a question, and I'd be happy to take a moment and address that. All right, so this is what we did with the screening, male and female CD1 mice, and these were our first set of data from this. This is all generated by Adrian Pena, who is now graduated and works for Evident. but was working on his PhD with me and was the grad student foolish enough to say, hey, do you want to screen some completely unknown receptors to try to find something that might be active in chronic pain? So he said yes, which worked out this time. But if you're a student listening, maybe ask some questions if your PI approaches you with that kind of pitch. And so we have the CIPN. So you have the pre and the post CRISPR baseline measurements, the post CIPN measurements. So that the mechanical sensitivity is greatly increased, the threshold goes way down, and then you inject them with, in this case, the morphine, and they get less sensitive for a while. So this is sort of that basic shape of the pain relief von Frey filament testing in CIPN. And as you can see, with 141 and 150, there's no impact at all, either on pre and post baselines, on the post CIPN baseline, or on the analgesic response. So these are obviously no effect, right? And so these are photorealistic representations of Adrian's face when he first got this data. He's very disappointed, very sad that these didn't turn out, but we turned that frown upside down for 63 and 153, which of course are the main title of the talks. You already knew that they were going to be the ones. But these now don't have an effect on the baselines that we could detect, but completely ablate the antinociceptive response. So this is very common with these models, as we'll see as we go along. It's not a like 10 or 20% reduction; it's 100% reduction, and we see that very consistently. So this was very exciting. We had some celebration and some excitement in the lab, but this is only the very first thing, right? So this is analogous to running a drug screen, and you get hits that light up your plate, starry night effect. But that's only the first step. Maybe your drug is off target, maybe whatever. Same thing here. Maybe the CRISPR is off target and so on, especially because these are orphan GPRs with really no established knowledge or tools to target them. We were really the first to do so. And so first thing we got to do is validate it, make sure this actually worked. And we can't validate this with immunohistochemistry, or we couldn't at the time, because at the time there were no antibodies for these GPRs. Again, unknown targets no one's working with. Exciting, but limit your options sometimes. And so we got RNAscope up and running to evaluate this in the spinal cord. This is the negative control CRISPR. You can see in red are your puncti for your RNAscope hits, in this case on GPR63 and down here on GPR153. And then you look over here with the CRISPR treatment, there's a little bit left in 63 and really none that you can really see in 153. So you get a very effective knockdown, at least of the mRNA. You might say, well, what about the protein? Well, we can't test that, or couldn't test that at the time. Now some of these tools are starting to come out so we can address some of these questions, thankfully. But we got very good knockdown. Like, okay, that proves that the CRISPR knocked it down, but what if you did some off-target thing and hit something else? So we wanted to, again, validate this. We needed to do a lot of validation because of the novelty of these targets. So we used a different approach using a small siRNA, again, complexed with in vivo transfection reagent, injected intrathecally. So sort of the same approach, but rather than CRISPR gene recombination, we're here doing siRNA-mediated mRNA degradation. So different approach, should be different off targets. And if the two agree with each other, that's some validation for you. And sure enough, when we did this testing, we saw results that looked essentially identical to the previous in the CIPN model. No really impact that we could see on the baselines, but the antinociceptive response was fully ablated with both the siRNA. And then also we validated the validation, looked at the mRNA levels by qPCR, and the siRNA was effective in both cases in reducing whole tissue mRNA for these targets, reducing them down about 50%. Okay. So we got some validation. That's good, right? We have some confidence that this first pain model has some impact, but that's only the very beginning. Just like this, literally just the first step that we had taken. So there's so many other questions. And the first real question that we wanted to ask is, okay, CIPN, great model, we love it. What about other pain models? And so the next pain model then we looked at was a post-surgical paw incision pain. And again, this is an audience that knows a lot about pain, so I don't want to belabor this or go through it point by point. But versus CIPN, this is a localized incision. Does have some pathological elements to the pain state. There is some, of course, local, but also central neuroinflammation. Some of these things kick in. But unlike CIPN, this is a relatively quickly recovering pain state. So within a few days, they kind of were back to normal, at least by von Frey measurement. And then again, we also there's a different timeline on this paw incision. It's not an 8-day thing like the CIPN. So we interwoven it with our CRISPR approach. So again, the CRISPR starts on day 1. You have these 3 IT injections, day 1 through 3. You do your testing on day 10. But with our paw incision surgery model, this typically we always measure 24 hours post. And so on day 9 now, we did the paw incision surgery. So that day, 24 hours later, after the paw incision and 10 days later from CIPN, those coincide. And then we do our testing, inject a morphine and run the time course. And then we also get another advantage from this post-surgical paw incision pain. It's sort of a value add that we wanted to look at. So of course you have the immediate pain state the next day. You have their response to analgesic drugs, which is all very interesting and important. But as I mentioned, over the next few days, they recover. And you can see this recovery quite nicely. Their mechanical thresholds kind of come back up. We see about 50 to 60% recovery in about 2 days, so about 72 hours. And by 4 or 5 days, we usually can't really detect much in the way of any sort of mechanical sensitivity. And so after this morphine treatment, we then looked in the next 2 days post-surgery and just took recovery baselines. And one thing I want to emphasize here is there's no injection, no drug. So just looking at recovery alone, no injection of morphine or any other antinociceptive. So what does that look like? So first of all, 63, no effect, which was interesting. Now we're starting to differentiate the receptors from each other. The 63 had no effect. 153 had a really interesting effect. And so first of all, that 24 hours after with the morphine injection, we're losing the antinociceptive response, almost as strongly as what we saw with CIPN, about like 80, 85%. And so that is sort of further shows in this additional pathological pain model that we have this effect of GPR153 and antinociception. But something that was really exciting to us is that when we looked at the recovery, we also see a difference. So in the negative control CRISPR-treated mice, you see they're kind of coming back up. They're about 50% or 60% back up to their baseline by 72 hours after the surgery. But the CRISPR, the GPR153 CRISPR knockdown, way less, right? So these are not really recovering so well. Again, no drug involved. This is just recovery. And so this long-term could be really its own sort of project. So it would suggest that GPR153 not just involved in pain response and antinociception, but also resolution of the pain state and potentially recovery as well. So this is something we'd like to dig into in the future and are pretty excited about. All right, so as I've been pointing out to you as we've kind of been going along, we couldn't really detect any effect of the CRISPR on the baselines here after the CRISPR treatment. Can't really detect any effects after the pain state initiation, although they're very, very sensitive, so it could be hard to detect if they made them pronociceptive. And where we're really detecting the effect for most of them is in the morphine response, right? The antinociception. But as was helpfully pointed out by some grant
John,
reviewers, not that.
sorry to interrupt, John. Sorry. Thank you. I had a quick question of how
Yeah,
long is the CRISPR effect good for? I don't know if you mentioned that already or not, but relative to looking at the results later for your recovery. Thank you.
yeah, absolutely. The actual modification is permanent, and then but we've never followed it out long enough to see what would be sort of that recovery. And so we know that it's good for at least 2 weeks, and we haven't had a model that's gone beyond that.
Great.
When we
Thanks.
do, we'll want to test that.
Yeah, got it.
My suspicion is that it's very long term, like months, maybe longer, but we haven't actually tested that. All right, great question. All right, so we have this effect where it's not blocking not the pain state it seems, but rather the antinociceptive response to morphine. But as was helpfully pointed out by some grant reviewers, like if this is opioid selective, then the impact and sort of implications of this as a novel regulatory system are greatly reduced, right? So if it's sort of opioid specific, now everyone's, oh, I don't know, this, opioids, no one likes opioids. Let's run away from this project. And so one of the things we did, especially as we're building up into our resubmission of the grant, which just went in a few months ago, is to ask this question about other analgesic drugs besides opioids. And so we did. We looked at them and found that the effects are not specific to morphine. And we looked at 2 drugs. We looked at gabapentin. So again, you all are very knowledgeable, but the presynaptic voltage-gated calcium channel inhibitor, as well as WIN55212, which is a relatively non-selective synthetic cannabinoid agonist. Both produce antinociception, as you can very clearly see very well in the CIPN model, neuropathic pain. That's great. And then in all cases in the CIPN model with both GPR63 as GPR and GPR153 with both drugs, you see that replication of that effect that you saw with morphine. So complete loss of antinociception. So it's not just morphine, not just opioids. And then we're also replicating this with GPR153 in the paw incision. Gabapentin, not as effective, of course, in the paw incision model as a neuropathic pain model, but we see that same effect here. And then we have one cohort done and one more to go to replicate this for GPR153 with the WIN. So that's coming soon. But this is important, right? So this suggests that the effects are not opioid specific. It's broad and not even necessarily receptor broad, right? So you might say, well, the opioid receptors, cannabinoid receptors, G-alpha I coupled mechanism, kind of similar, blah, blah, blah. But gabapentin, of course, is a very different drug class from either of those, from opioid or from a cannabinoid. So the effects seem to be fairly broad. And then building off of that, so we tested this in CIPN, pretty strong pathological pain model, the paw incision, still pathological, but not quite so strong. And then we start to differentiate between GPR63, GPR153. So then we wanted to say, what about a non-pathological or uninjured pain state? And so for this, we took uninjured mice, no surgeries, no paclitaxel, nothing like that, treated them with the CRISPR, and then this time using tail flick antinociception. And then because they're uninjured, we just do that on day 10. Like we don't have to have any sort of blending of timelines here. Still inject them with morphine and look at their time course, but this is all tail flick in uninjured, non-pathological mice. And there, no effect at all of either receptor. So now it really seems to be building towards that these are, at least so far, seem to be somewhat selective for pathological pain states. Even though the effect is on antinociception, it's sort of selective for pathological pain instead of a non-pathological, uninjured sort of baseline state. And there seems to be a strength effect where, starting with CIPN, which is the most pathological, neither receptor, then you go down to paw incision, which is sort of in the middle in terms of pathology, and then GPR153 now is the active one, and then you go to tail flick, and then neither are active, as it's a non-pathological pain state. And so we don't fully know the basis for that yet, but it's exciting, and we're continuing to push forward on that study. Okay. So in these studies now, we've gotten at least a feel for the breadth of pain that they're active in, right? First, I mean, this took a long time just to get to this point, and it is sort of initial beginning steps, but we have some idea now that they seem to be active in pathological pain states. But even then, there's some questions remaining, and one that Adrian and I kept talking about, what is actually being regulated? Is it the pain state itself? Are they decreasing the effect of CIPN or paw incision or whatever, or increasing rather when you knock them out? Or is it selective to the antinociceptive? And one of the things that was preventing us from detecting that is the way that we're doing our von Frey is very good for detecting antinociception, but the sort of the up-down method with the finding the active filament, there's no room here. They're so sensitive at this point with this method that there's no room for a pronociceptive effect. There's only room to observe an antinociceptive effect going up. And so we changed our von Frey method to sort of test this in a set of experiments. Instead of using a series of filaments with the up-down method, we used one filament applied 10 times. And then instead of recording response, we record the number of responses to that same filament out of the 10 as a percentage. And now our baselines are shifting. They're not way down at the bottom. They're moving up and allowing us to more easily detect this effect. And so GPR63, GPR153, they look the same. You have your pre-CRISPR and they post-CRISPR. They're responding some, but not a huge amount. After CIPN, they respond more. So this is all consistent and sort of validates that we're doing the method correctly. But the knockdown had no effect, right? So the CRISPR, the red and the green, there's zero impact here on the nociceptive thresholds. So at least in our experimental models that we're testing so far, keeping on digging down, keeping on figuring out the details of how these receptors seem to work, it seems to be selective for the antinociception versus the actual nociceptive thresholds and the actual nociceptive pain state. At least that's the suggestion. Again, we're still sort of digging in and figuring out what this mechanism is, and it's going to take a lot of time to fully understand what these are doing, but we're starting to build a path. All right. Another question as we're going through this. We wanted to know if pathological pain regulates the receptors. Reason for that being, you have no effect in the tail flick you have a strong effect in the CIPN. Maybe that's because the pain state is activating those receptors, turning them on. And that's not completely out to lunch or completely unprecedented. From a different project that we're working on with the A2AR, this is in the spinal cord, dorsal horn. I think you probably all know what that looks like. We get an extremely strong upregulation of this receptor in CIPN. And so we said, well, maybe something similar is going on with the GPRs. But of course, we don't have antibodies. We didn't have antibodies at the time. We weren't able to test this with a very nice immunohistochemistry like we did for the A2AR, so we had to use qPCR. And we actually did see something though. So 63, no effect, but in 153, you see this upregulation in CIPN, now suggesting that perhaps they are responsive. Maybe at least the 153, maybe it's being turned on. I mean, that's very sort of a not well supported conclusion at this point. There's a lot more work to do, but this is at least a clue that sort of points in that direction, that this could be being turned on during pathological pain. Okay. So again, sort of taking stock, what we've done, where we're at, we seem to, by looking at these different pain models and sort of poking around what the pain aspects are, we seem to suggest that these receptors are suppressing pathological but not nociceptive pain, or at least pain relief. That we're sort of at that point, but of course, nothing known about the mechanism. And there truly was nothing, again, nothing from the literature. It's like, so that we think they're GPCRs, they've got the right structure for it, the right sequence for it. We don't actually know that. Like, no one's actually expressed them in a cell and studied them and said, these are receptors, these are not receptors. And so we think they're GPCRs, but we know nothing else. We don't know what G subtype they couple to, what their cellular activity and signal transduction cascades. We don't know any of that. We don't even have a basis to even speculate on, or we didn't at the time have a basis to even speculate on what the mechanism is. And we didn't even know where the receptors are expressed other than in the tissue, right? So it's very grind and bind style analysis from the GTEx and these other proteomic and transcriptomic data sets. So we wanted to know this. And again, we didn't have receptors, or sorry, we didn't have antibodies. And so we had to use RNA-scope again. And so here is one example image, GPR63. We're zoomed out a bit so you can sort of see the shape here of the dorsal horn, right? So these are sort of your superficial layers. These are sort of some of your deeper layers. And just looking at it, you can kind of see that GPR63 is, I mean, this isn't quantitative quite in that same way, but seems to be fairly highly expressed and fairly broadly expressed across the gray matter of the horn, right? You're not seeing so much in the white matter, but in the gray matter here, it seems to be pretty strong. But that doesn't tell us anything. It doesn't tell us what cell type they're in or anything else that's going on. So we needed to co-stain. And here we co-stained with AIF1, which is the gene name for IBA1, i.e. microglia. And we did the same thing with GFAP. And you get the puncti, and then you look down here and you're like, oh yeah, some of these, it looks like they're co-localized, right? You're getting some green and red that kind of look like they're in the same cell. Now, it's not quite like IHC, where green and red makes yellow. They don't usually overlap in that way. These are different granules within the same cell, but they don't directly overlap usually. And so you have to find out if they're in the same cell. And that's not so easy as just obviously looking for yellow. You don't really see any yellow when you're looking on here, but it does look like these are in the same cell. So how do we analyze that? How do we tell what's going on? So we use segmentation analysis with QPath. And so basically what that does is, here we have the DAPI, you have the blue, and the software very good at detecting and defining DAPI. It's a very clean signal. And the software is basically making the conclusion every DAPI is a cell, and then it segments it is somewhat arbitrary, but segments a cell around that and sort of how they fit together. And you can sort of see it's drawing cell boundaries for these different cells. Even when the tissue's packed in, it's drawing something that at least is a good guess, a good software guess at what a cell is. And then you look within that cell and ask the question, are there green granules, are there red granules, or are there both? And way zoomed in, you can see examples of that. So here's a cell, you see the blue, you see a bunch of red, you see some green. This would have been counted as a positive colocalization cell, whereas this cell down here with just the green granules really not going to be colocalized, right? And so we use the software, do the segmentation analysis, do the counts, and found that both GPR63 and GPR153 are fairly strongly expressed in both astrocytes and microglia. GFAP and AIF1 is the markers. And so 63 is in about 50% of both, 153 in about 50% of astrocytes, and kind of remarkably, almost all microglia, at least by this analysis. Seems to be very strongly associated with microglia. Maybe that's why the 153 had the effect in the paw incision and the 63 did not. I don't know. We're still sort of figuring some of these things out. So we have, for the first time, some suggestion about where these orphan receptors are located, what cell types they're in, but that doesn't, of course, tell you anything. That doesn't link it to the pain state. We had to know which of these cell types was involved in the physiology, the impact on the pain behavior. Was it astrocytes and microglia? Was it both? Was it neither? We needed to know that. So how did we test that? So I sort of alluded to this earlier, but we've developed this CRISPR technique to be cell type selective. How do we do that? We take out this CMV promoter that's driving the Cas9 protein cassette, which again is needed to drive the genetic recombination. We take this promoter out and we replace it with a cell selective promoter. And so for this particular study, we use AAF1 to drive expression in microglia, GFAP to drive expression in astrocytes. We've also done this in other papers with CGRP and other markers. It works pretty nicely. It's a very effective approach. And then look at this behaviorally. Transfect these CRISPR constructs just like we did previously, except instead of tissue knockdown, it's for these cell selective knockdowns, and then look at the effect. And we saw the same thing in all the testing, both the CIPN, 63 and 153 in CIPN, and 153 in paw incision. Black is the negative control. Of course, that's responding like normal. And astrocyte, the magenta, no impact, very much just overlaying the negative control, even here in recovery. Whereas the microglia completely recapitulates the effects of the broad tissue knockdown, including on paw incision recovery with 153, right? So it's doing that too. And so, I mean, there's still some questions. I don't want to be stated too strongly, but this is pretty solid evidence then that although the receptors are expressed in both astrocytes and microglia, at least in terms of pain and pain response, the microglia seem to be where it's at. Like that is this probably the site of action through which they're carrying out their role. Okay, so we got this. Of course, this was very exciting. But then we got this, and then we started thinking about, okay, we know almost nothing at this point. We know that these receptors are, we knock them out, we lose pain relief, and it's broad pain relief, and it seems to be the microglia that are the site of the action. And there's no information on these receptors in the literature. But of course, there's a lot of information on microglia in the literature. And sort of by a little bit of deductive reasoning, we're able to sort of assemble a preliminary model for what we think is going on, which we would then go on and test further, right? Of course, we always test our models. But what we think is going on, you knock out GPR63, GPR153, you get an decreased pain relief. And then we know that's being carried out through microglia, and we know that microglia increase pain and decrease antinociception by the secretion of cytokines. The active form of the microglia does that. And so sort of putting that together in a rational deductive way, we concluded that 63 and 153, when they're present, when they're not knocked out, are a negative feedback loop that's pushing the microglia back, pushing them back into the inactive state, making them angry, or starting out they're angry, they're trying to make them relaxed and chill and put their arbors back out and shrink down the soma, stop secreting so many cytokines, stop being so upset. And that is sort of what we infer just from the results and what we know about microglia in the literature. So we think they're pushing them back here, blocking the active microglia state, presumably blocking the secretion of cytokines, and thus promoting pain relief by now multiple classes of analgesic, opioid, cannabinoid, voltage-gated calcium channel at least. We haven't tested others. So this is the model. Great. Now let's test it. This model has some suggestions. This model would start off by suggesting that when you take these out, 63 and 153, when you knock these out, you're going to get more active microglia and more cytokines, and that's blocking the pain relief. Okay, that's the prediction. Let's test it. And so we first tested this by injecting minocycline. So minocycline is a microglial, at least polarization inhibitor. I don't know how everyone falls in the M1 versus M2 thing. I don't want to start any fights, but certainly seems to decrease the pro-inflammatory activity of microglia, right? So we do the CRISPR, we have the CIPN, and then right before we inject the opioid, in this case, we inject the minocycline to disable those active microglia. And so here you have your negative control in blue. In red, we have the knockout with vehicle pretreatment. This is exactly what I showed you previously. We get this blockade of antinociception. However, in green, when we inject minocycline, that perfectly recovers this knockdown. So all we do is inject minocycline, and we go from here back up to normal. Same thing with 153. Exactly the same. Very clean response, very clean data. And so this is now some more direct evidence that the microglia are more active when 63 and 153 are knocked out, suggesting that when they're present, they are deactivating them, suppressing their activation. Okay, this is a good experiment, but minocycline is dirty. It has off-target effects. Let's keep going. What else can we do to sort of prove that this is what's going on, or just support, provide information or evidence that this is what's going on? So with the help of my colleague, Laurent Martin, who's a very talented faculty here at the University of Arizona, collaborates with us a lot, very good guy. What he's been working on is this microglia morphology modeling. And so, we're all familiar. You take the 2D slice, in this case, stained for IBA1. You can see the little microglia, like we kind of know what that looks like, right? However, if you take a stack and then use some very nice software called Amaras to analyze that 3D image of that cell in a stack. So now you're not just getting the optical slice, you're getting the whole, presumably, cell. You can reconstruct a 3D model of what that cell looks like, and that's sort of shown here. So this is the 2D slice. We take a stack and then use that to build 3D model. And okay, let's look at these 3D models. So this is the negative control CRISPR with CIPN, so they are inflamed. We're still doing the other controls to fully flesh out the set. But in that circumstance, you can see you've got a microglia. It's got a soma. It's a bit condensed compared to what it would normally be, but you can still see some of the arborization and some of these things sticking out here. However, when you knock out 63, you knock out 153, you can visually see this, right? Like you look at these two, you look at this one, you can visually see the simplification and the condensation of that arbor, the increased size of the soma. These are hallmarks of microglial activation. Here's a quantitation for that. Very nice, clean, highly significant quantitation for both the soma volume and the process length. You do that knockout, it's producing a morphology that is much more consistent with microglial activation. Okay, so those are then two pieces of direct evidence, but there's more that would still need to be done. And so, we have now some evidence of sort of how these receptors are interacting with the microglia. The very next thing we're testing, like literally right now, are the status of these secreted cytokines, which we predict will be enhanced by the knockdown of these receptors. I took a point of personal privilege because I'm so invested in this project. I'm actually running this experiment myself, which I very rarely do. The mice are going to be ready this Friday in 2 days, and I should have the data next week. So wish me luck, fingers crossed, that we can directly show that the cytokine levels will change. But we now have direct evidence to support this model that these receptors are acting as a negative feedback system to take the active microglia and push them back towards the inactive, or at least prevent them from making that transition in the first place. Might perhaps be the more accurate way to say this. And so these are kind of early days, even with all of that work, which is taking years to build. Like there's so many questions that you probably are thinking or want to ask about all the additional evidence that needs to sort of be generated and shown. We're pursuing that, but so far right now, this is sort of where we stand in terms of identifying these as novel GPRs in the spinal cord, novel role in regulating pain, and sort of this novel effect on regulating microglia as new microglial regulators. And I very much hope to get the chance to complete that project, assuming that we can get something funded through the NIH, which is very tricky right now, as you all know. A few additional points, though, before I wrap up. So one thing you might ask is, okay, with this experiment, you directly show that this knockdown is changing the microglial morphology. That should be consistent with an altered cytokine profile, which I expect to find when I run that experiment myself next week. And you're like, okay, then why does it not impact the sensory thresholds? Why does it only impact antinociception? Well, what I think is going on, and there's evidence both from my work as well as, of course, much more evidence from the literature that suggests that there are different thresholds of sensitivity for microglial modulation of nociception versus antinociception. Nociception, harder to do. Antinociception, easier to do, more sensitive to changes in microglial state. If you all want to ask later, I can sort of point you towards several papers. But one example would be we took in a different publication for a different project, we're still nonetheless focused on microglia with the two different microglia inhibitors now, minocycline and PLX3397. In this case, these mice do not have CIPN. They are uninjured. There's nothing wrong with them. Vehicle here in red, and then open red is treatment with PLX or minocycline. This enhances antinociception uh, in uninjured animals, all on its own. Again, not selected for 63 and 153, but we can show that you can modulate this antinociceptive effect in uninjured animals at baseline by just injecting these microglial inhibitors, whereas that does nothing here to the baselines, the nociceptive baselines, only the antinociceptive. So I think antinociception is more sensitive, and we'll probably be able to dig that out and figure more of that out as we go along. And then, of course, as I mentioned, there's so much more to do. I'm measuring those inflammatory cytokines. We need ligands. So this has all been done with sort of knockdown tools, right? So all we have at this point are nucleic acid tools. We have no ligands, exogenous or endogenous, which is challenging because we have an assay an in vitro assay to do drug screening, which we're working on. But how do you validate that assay? There's no drugs. So it's been an interesting and exciting challenge. But we need to find those ligands. And then, the endogenous ligands, which I predict are going to be upregulated in pathological pain states to stimulate these receptors as a negative feedback loop. And then, of course, mechanistically, there's so much more that we need to find out, particularly after we get these ligands. I want to know what they're doing in microglia and mouse spinal cord in terms of their mechanism, what they're doing to the actual microglia, how they're changing it. I'm sort of hypothesizing, very early hypothesis, that they are G alpha I coupled to achieve this effect. That would kind of make sense, but we don't know. We have to figure that out. And then, of course, other diseases. So this was all just in the context of pain. But in principle, if this is a broadly acting neuroinflammatory regulator in microglia and maybe astrocytes, there could be a lot of diseases that this is involved in. Neurodegeneration, even addiction, which has neuroinflammatory component to it. Maybe even aging, which has been something I was just kind of throwing out there in the lab, since aging has a neuroinflammatory component. And then what are these receptors doing in the other cell types? So I showed you that they're expressed in in astrocytes. There's plenty of them there. What are they doing there? What's the physiology? That would be great to know, as well as, of course, as neurons. These are all things that I really hope to be able to spend the next 20 years digging into. We've made a little bit of progress on some of these fronts. So this is some of the very, very early, very preliminary ligand screening that we've done. On top is an FDA library screen for 63 and 153. I just told you that we don't have a way to validate it, but nonetheless, we got some hits. And so we're following up on these now. Hopefully we could show that they're bona fide exogenous ligands, which would be very useful to validate as well as test in vivo. And then in terms of screening for endogenous ligands, we took spinal cord extracts and started testing these fractions in vitro. And we got a hit, at least it looks like a hit, on one of those fractions. So now we're running mass spec. We're going to follow up, and hopefully we identify an endogenous ligand, which would be very exciting. So these are sort of the summary messages. Both of these orphan receptors, 63 and 153, we have evidence to suggest they are novel regulators of pathological pain specifically. We think they're acting as a negative feedback loop on microglial activation, suppressing microglial activation, pushing them towards inactivity. And then we think that here that 153 sort of has an additional role, maybe because of its higher expression in microglia, in post-surgical pain, antinociception, yes, but even more importantly, on that recovery. Which is not drug involved. There's microglia have a very, this isn't my area of study. I don't want to step on anyone's toes, but microglia have a well-established role in pain resolution and recovery. The secretion of resolvins, like all these factors that get secreted. I predict that 153 is involved in that, is involved in regulating those recovery and resolution states in microglia. And I dearly hope to be able to study that. And then, of course, take-home message. These are orphans, so there's just so much more to do. And these are the people that made this possible. These are the folks on the GPR project itself. I particularly want to highlight Adrian, who took that leap into the great unknown with me and did most of the initial work to find this. Other members of the lab not on this project. My collaborators, Chris Cartmel, very important on this work. Laurent Martin, even more important in terms of helping us do the microglial biology. David Min at IIT has been helping us on computational modeling to hopefully find a way to computationally screen these receptors, and then of course our funding, and then I have some conflicts as well, although these are not really directly relevant to this project. And so thank you again. I greatly appreciate the invitation and the chance to talk about this work, and I really hope to take some questions and talk about this more.
Thank you, John. That was awesome. Well, we could, first open up for questions from the attendees. Well, I'll start. So given the widespread, I don't know if you want to stop sharing for a minute so we can all see each other. Um, there we go. That given the widespread expression, of course, as I recall, one of the RNA scope where you focused in on the dorsal horn, of course, it seemed like there was a fair amount of staining in the ventral horn of this cord. And I was just curious. I mean, I understand that the Von Frey testing, of course, at baseline didn't seem to change, but
Mhm.
have you thought about potential confounders, as it relates to on the efferent motor side of this with your behavioral testing and or future experiments?
Yeah, absolutely. So we haven't run the rotor rod on this one, and the easy one to do.
Hmm.
And then I'm very, in that sense, I'm very heartened by, say, the response of the tail flick, right? So this is clearly not
Hmm.
a universal loss of motor function. It's, as you just mentioned, the thresholds are fine, they're unaltered. But then, even in, say, the tail flick, the antinociceptive response, which requires a rather large motor response, is normal. And so, at least so far, that would suggest. But yeah, we got to look at these confounds, and looking at, say, an affective model of pain measurement, right? So my colleague Frank Pareca, of course, has made very important contributions in that in using condition place preference. We'd love to be able to do the same. To sort of show that this is affectively, reflected in the affective pain state. We now have a home cage monitoring set up. I want to look at pain suppressed behavior, eating, grooming, feeding, social interaction, all that stuff. We have that system it's set up, so 100% we're going to do that. Part of that grant that was just submitted. And so, yeah, it's like I definitely I'm concerned. I mean, so far there's no sign of it, but we're always going to be concerned about that and try to do these experiments to really show that it's the pain state being altered and not just, messing up their motor function or whatever.
Great. Other questions from the gallery this time? I had another question if no one's coming forward, and that has to do with its relative distribution in the brain, central nervous
Mhm.
system, of course. You initially laid out the strategy of selecting, candidates based on their co-expression in areas that have been identified in the pain pathway, as it were. Um, does that, so how do you loop back to that? I mean, I know there's the focus on the dorsal horn and nociception, but relative to these global effects of behavior on pain responses, I was wondering if you've done any additional work on fine mapping or even just some initial RNA scope in brain regions that have been previously studied and involved.
Yeah. Yeah, that's a great question. No, we have not. So that is definitely on the docket, right? We got to go in the brain.
Hmm.
I would love to, and we're gonna at some point, assuming that I can get a grant.
Oh yeah,
So
understood.
we'll
Yeah.
100% do that. But again, like there are these atlases, right? So the human
Hmm.
protein atlas and the GTEx data set and so on. And so there are regions, like they've taken the whole region
Hmm.
and looked at expression, but no one has done fine mapping, and we haven't done it yet. And so doing so, very informative, and I definitely hope to get the chance to do so.
Great. And I have one final question, and it seemed to be hogging the microphone here, but this has to do with, of course, since we're also working on an orphan receptor for a while, defining distribution of expression and other aspects. But circling back to the ligand search, right, is a tricky business, either using indirect, coupling assays and doing library screens. Since you're in on it, not to try to reveal any secrets in the lab, but just general, approaches. What are your take-home messages so far for when you have essentially an interesting orphan molecule, but you really have no idea of its ligand? Do you think
Yeah,
grinding up and reapplying versus screening libraries, do you have any guidance or hard-fought wisdom around that?
absolutely. So it is tricky. Like even you think about, decades ago, and the typical way, say, of screening opioid ligands was to do guinea pig ileum or mouse fast deferens. Like you sort of had these tissue bioassays, right? And people weren't really doing like G protein coupling in a cell line yet. But you had ligands, right? So you could validate the assay. There is no bioassay for for these receptors targets. We'll probably identify one as we go along. So you can't really do that. And so there are tools available, of course, like you can make any construct that you want that you could turn this into an assay, at least in principle. And for our efforts, we took advantage of the Presto Tango system. So for those of you who don't know, Brian Roth and his group published a wonderful paper a few years back where well, more than a few actually now, but they took almost every non-olfactory GPCR and turned it into a Presto Tango construct, made that publicly available at Adgene. And
Mhm.
so you can just go buy it, like $85, I think they're at now for an Adgene vector,
Mhm.
and just go buy it. And that's what we did for 63 and 153, both of them, as well as some others. And so we can get the assay and get it set up. But then in terms of like, what do you do after that, right? So like we can't actually validate that it's working properly. And so we've just tried to take a broad multifunctional approach to sort of try numerous or multiple things at the same time,
I
thinking that
see.
if one
Yeah.
doesn't work, one probably will. And so in terms
Sure.
of physical screening, yeah, absolutely. We've got the Presto Tango. We're
Hmm.
also working on TruePath to get a TruePath construct so that could just be a side by side, a different approach. And then, we did the FDA library screen. We're working on natural
Right.
product screens. Like we're working sort of multiple chemical sources, right, to do these screens that are feasible,
Mhm.
that aren't going to cost
Yeah. Right.
$50,000 per run. And so
Exactly.
sort of doing that in multiple ways in a physical sense, but then
Mhm.
also at the same time, like maybe these assays aren't working right and we don't know it. So let's do the computation. Let's use AI to the extent we can. So
Mhm.
we're talking with collaborators about trying to make crystal structures of these so we can do this properly.
Mhm.
But for the moment, we've made AlphaFold predicted structures. They
Mhm.
do look like a GPCR and use that to start to do computational, to start to predict molecules that, and hopefully we could easily make. Like, so you use like an
Mhm.
existing compound library instead of a theoretical compound library, something where you can sort
Sure.
of accessibly access that chemistry.
Mhm.
And we're running the computation and testing those. But even within computation, we're doing multiple methods. So we're doing a more with one collaborator, Fahad Al-Bayadi, we're doing a more traditional sort of static docking with peptide ligands. And then with David Min, who I mentioned, a different
Mhm.
collaborator, we're doing a molecular dynamics simulation based approach with small
Mhm.
molecule libraries. So like all these different complementary but
I see.
different approaches
Mhm.
to hopefully get one that'll work.
Awesome. That's great.
Don't put all your eggs in one basket. I guess that's the
Yeah, I
wisdom,
think that's the take-home
the
message.
cliche
Thank
wisdom.
you. Perfect. Well, fantastic. Well, for Park and the rest of us, thank you so much for this excellent seminar and lots of work ahead, but exciting work. So thanks again, John, for your time.
Thank you, all of you, so
Yeah.
much. I really
Yeah.